Okay, this is Tom Stark. This is the NAMI Santa Fe Public Information Talk Series. Tonight we're very fortunate to have Dr. Nora Perrone-Bizzozero from the UNM Department of Psychiatry, who will be talking on autism.
Well, thank you, Tom. Thank you for the invitation. This is my second talk, and I'm delighted to come back. For those that heard the first talk, I just want to give you a little bit of information where I come from. So my training is from the University of Buenos Aires, Argentina, where I got my PhD in biochemistry, and also had genetics training there at the Human Genetic Foundation. We moved to the States, and I was a postdoc in the Department of Psychiatry at Harvard Medical School, and then I moved to UNM in the Department of Biochemistry and then Neurosciences, and now I have also an appointment in psychiatry. So my research interests are primarily neuropsychiatric disorders such as schizophrenia and autism. I'm not a psychiatrist by training, so my specialty is more looking at the genetics of mental illnesses and trying to understand the causes as well, the genetics and also environmental or different toxins that can lead to risk factors. So for tonight, I'm going to talk to you about the genetics of autism spectrum disorder, and we are going to I'm going to talk about the genetics and then the brain circuits. And finally, I want to leave in a really high note. Since this year, there are 2 new drugs that were approved for the treatment that show really very promising results. So to start with, I'll start with what is autism. And autism spectrum disorders are characterized by several symptoms. The 3 more important is impaired social interactions, delayed language and verbal communication, and then stereotypical repetitive behavior like rocking. Children will have other types of what is called steaming, hand waving. They will have several sensitivities to sound. They could have, I don't know if I can use the screen, so sensitivity to sound, really passionate interest, repetitive behaviors with the hands, and social confusion. So it will be hard for them to understand what others think, so interpret human intentions, and this is critical. As a society, we need to know what other people are thinking and feeling in order to communicate effectively. So this is basically the hallmarks, the main hallmarks of autism. The types we have, the classical type, which is the one that is more prevalent and usually is diagnosed before 30 months of age, and this is called autism, or also within that spectrum, and this is from the Diagnosis Statistical Manual 4, and I'll tell you what is the difference now in the Manual 5, Fragile X syndrome, that it will be separate, and I'll talk at the end briefly about Fragile X syndrome. Then we have the high functioning autism group, and then we have this pervasive developmental disorder, also called otherwise specified, PDD and OS, and this is in the DSM-5, now Rett syndrome, which I also talk, those are my last 2 slides, Fragile X and Rett syndrome, are going to be in a separate, and I forgot to say Asperger syndrome is really the other high functional autism group that is also now in a separate entity. And then there are other childhood disintegrative disorder or CDD that are very, very rare. So the most common is the autism, the first group, and I'm going to start with that telling you about the incidence. And if you remember when I gave the talk about schizophrenia is 1% of the population, and this is very prevalent to also 1% of the population. And in schizophrenia is more or less globally is the same, but in terms of autism, depending on the diagnosis. So some groups will vary the prevalence, and this is not because autism is different, but how it's diagnosed by the clinicians. So it could be, the estimates by the CDC, so in the US now is 1 in about 58, 59 children, and then the other subtypes that I was telling you, Asperger and is half of that or a third of that, and the other forms are even more rare. So basically, this is a highly prevalent disorder affecting children. But it's not the only disorder affect children. And I forgot to say that they have affects mostly males. So 4 to 1. So every 4 children affected will be males to 1 female. And it's not the only. Do you have a question? Sorry. Okay. So it's not the only disorder, and we know that there are more prevalent disorders in children, which call mental, behavioral, or developmental disorders as a classification, and affect about 1 in 6 children. And this is due to other factors like poverty, lack of health care, abuse. There are many, many causes for disorders in children, and this one is very specific. Autism is separate from the other type of disorders. So as I don't know how many of you were here last time, but we talked about schizophrenia as being very hereditable with 80 in twin studies. They have this meta-analysis here, and you can see it's 81%. So it's very highly hereditable. But in Autism Spectrum Disorders, it's also true that we have the same. It's more or less the same, starting from the general population of 1%, going through how much is of the genes are shared. So going for cousins to siblings to dizygotic twins, and finally a monozygotic twins, which are identical genetically. The range of incidence will be between 40 and 90%. So this is, again, very prominent genetic risk factors in there. And I'll talk about the genes in a minute. But first, I'm going to go over again some of the definitions that we went last time, just to be sure that we are on the same page. And okay, so genetics is the study of variation, hereditability, and variation in inherited characteristics. It can be from eye color to intelligence. So there are many traits in the population that, like height and others, that are hereditable, and they have schizophrenia and autism are also highly hereditable. Then what is a gene? So we talk genetics is the study of genes. So a gene is a functional unit that characterizes a trait, and in this case could be a protein or a series of proteins. So a gene is a protein, but we now know that there are genes that don't encode proteins. They are not just all the proteins in your body. They are also regulatory genes that we are going to see in a minute. And the most important part, again, like we said before, we are not talking, we are talking risk or liability, meaning that there's, as you're going to see, there are thousands of genes that contribute to disorders. So no single gene is enough except the last two I'm going to tell you. But in the same, like I said before, in schizophrenia, it's 1,000 of genes with small effects, so they combine together. And in this case, it's the same. There are really thousands of genes, but there are a little bit different genes than schizophrenia. So we'll talk about what is common and what is different as I go along. So in terms of the human genome, we know we have 3 billion bases. The bases is base pairs of DNA. So this is our DNA. And if you can see here the DNA, in each chromosome we have a chromatid, and in each chromatid we have a strand of DNA. And that strand of DNA is where you have all this coding for proteins or others. And we know that we have 23 pairs of, and is 22 plus the X and Y. And so the idea is to understand more how all of these contribute to this. We need to know what is the source of variability in the population. And the main sources of variability in the human population is either the single nucleotide polymorphism, which in genetics we call SNPs for short. And to this example here, so you have, there are always, 4 bases, adenine, guanine, thymine, and cytosine. Okay, so this A, G, D, C. And what happens if this G becomes a T, then this sequence is going to be changed. And every 300 nucleotides, sometimes 500, we have natural occurring SNPs. And some SNPs, it doesn't matter because they are silent, and some others have some important functions. So, but that's those are so there are millions of SNPs, and as I'm going to say, we have ways to analyze SNPs in each person using arrays or using DNA sequencing. I'll tell you more about it in a minute. The second important changes that is different between all of us is not only the sequence of the DNA, but the, in terms of the sequence is the what we call copy number variance. And copy number variation means that its sequence is either present and duplicate or is absent. So it's a copy. So it's either inserted or deleted. So it's duplication or deletion. And as we're going to see in next, we can look at schizophrenia and autism in the terms of SNPs and CMVs. So let's move on to where we left last time. So even though most of my talk was devoted to the genetics of schizophrenia, at the end I wanted to tell you that there were other genetic disorders like bipolar disorder as highly heritable, and also Autism Spectrum Disorders. So in those boxes I highlighted how much of the heritability of these disorders are due to common SNPs. And you can see that it's about 20% for schizophrenia, and it's like 15% for autism. But in this study by this cross-disorder group of the Psychiatric Genomics Consortium, which I introduced last time, which is a large consortium, 120 countries in different disorders, and all of these disorders are covered by, as part of this consortium looking for genetics of all these psychiatric disorders. They also look at comorbidity. So what is more likely in a family, or in a patient, or mainly in a family, if someone has bipolar or has schizophrenia, the chances that those are in the same family is 68%, and in this case, by the genes only will be like 15%. So that's. So if we look at the autism and schizophrenia, I know it's much less than schizophrenia and bipolar or schizophrenia and major depression, but it still is significant. So it's a 2.5%. So it means that some of the common shared heritability, they converge to the same genes and to the same pathways. Okay, so that's kind of the concepts, even if they are not exactly the same genes, will be pathways of brain development. And I'll go over this in a few minutes. So in terms of CMVs, and I presented this last time, but I want to reiterate that these copy number variations are present in schizophrenia, and this is a diagram of all the chromosomes in the human cells. And you can see in autism spectrum disorder is, sorry, I mislabeled them. Autism spectrum disorder is in red, and schizophrenia is in blue. And anything that is common in here will be in black. So you see there are some black boxes, but there are also red boxes, and there are also blue boxes. So we have a variation throughout the different chromosomes. So now I'm going to stop talking about schizophrenia. I'm going to focus mostly on autism and what are the sources that we know of mutation in patients with autism. And basically, as you can see in this pie chart, the 77% is unknown. We don't know why a child has autism. Then there are other causes that are in red that are called Mendelian single gene mutations, which I have a table. The third from the last slide, we have a table of all conditions that have autistic characteristics. And then there are chromosomal abnormalities and rare de novo mutation. But again, what we didn't know, and this slide is kind of old, but we know more now. This year, they have a study in which they show that unknown piece of the pie, what could be. And so this is really an important study, and it was done, again, by a large consortium. So you can see a lot of authors here. Each author contributes a different population of children. But you can see in order to do this type of genomic analysis, they needed 18,000 individuals with autism spectrum disorder and almost 28,000 controls. So you can tell, in order to do this analysis, because there are a lot of rare mutations that are less than 1% in the population, in order to detect that, you need a large number of subjects to do genetic analysis. And so how they did this study is the following. You have a large population of patients and a large population of controls, and you take the DNA and then look at the DNA for looking for the single nucleotide polymorphisms, and you analyze in what we call gene chips. And these gene chips had all probes. They have 5 million SNPs that can be read in this chip, and those are the common SNPs. But we can also, knowing the characteristics of the population, we can, using bioinformatics tools, we can impute up to 15,000 SNPs because of the way they are inherited as blocks. And so I went over that before, so I will not over that in this talk, but I can leave my slides again if someone wants to review the slides from before and here to kind of look at these sizes. And the important thing is the way these variations are. Either they are the major or most common alleles. So in this case, they call it AA. Then the heterozygous, you have one major and one minor, and the minor is BB. And the major allele is usually the ancient allele. So through these generations after generations, every time there are de novo mutations, so we get the main ancestral population is the major, the more frequent variant, and then as we go to the the minors are usually the most recent changes. And again, in this study, I'm not going to tell you all the analysis, but I want to say that all these SNPs that they found different, they all converge in neurons, the nerve cells that are important for the communication between neurons, and we think, see, so we perceive, we have emotions. All of our human characteristics depend on neurons. We can walk, we can talk, and that's sensory here, or seeing, vision, and all of that is due to neuronal communication. And in this case, they have identified new genes that were implicated in autism. Now, again, this was this year, and I don't know if you can see this table well, but basically all the genes in the table are expressed in neurons, and some of them are for a neurotransmitter or neuropeptide. Those are the way neurons communicate to each other, are proteins that are important for neuronal communication or neuronal development. So this is to tell you that this recent study confirms that most of the genetic variants associated with children with autism has to do with how the brain develops. And I'm going to show you some examples next, but this is from a previous study, so this is 2015. And basically at that time, all the genes variants that have been identified, or where they knew they were different in patients with autism versus controls, had to do with brain development, either before, I don't know if I can point, they are gonna see the screen if I point to the screen? No, okay. So basically, if you look at the age, in this first diagram in A, is the age, sorry, I need to go back. So maybe I cannot point, but I can tell you in the A, you can see how these genes or clusters, the genes were clustered in groups, and these are the most important groups. This all these are modules, those groups. And you can see that those groups are highly changed during development, either prenatally or postnatally. So you can see that the core here is first development, because we are talking about the onset of the disease and the influence of the disease being in the first year or two of life. The second, in B, shows what happens during normal development, and is how neurons are born, and then how they are making connections, and then they pathways myelinate. I'll show you how neurons connect through myelinated axons, and then which is the sheath that wraps axons to make information fast enough that is compatible with our human life. And then in C is where these genes are expressed, or what pathways. And the most important thing, I'll write it in the next slide, but in the brain we have the cortex, or the surface of the brain. So this is not my skull. If you were to open it and look inside, and after the meninges you will have the brain, and inside the brain, the top layer of the brain, the surface, is called the cortex, and that's where all neurons are. In the deeper parts of the brain is where the axons, they communicate one neuron with other neurons, and that's an important thing because we need communication between neurons to have effective brain function, okay? And then so you can see that the top layers of the brain is the ones that are mostly affected. And so it's a neuronal origin disease. And then there are other layers affected, and we'll talk more about this in a minute. So there's another very interesting study that was very recent. So this is May of this year. And they use single-cell genomics, which is looking at each cell and see what are the genes expressed in each cell. And what they found, and again, they use the cortex. You can see the little square in the top right panel. What it does, it shows you what piece of brain was used for this analysis. And you can see the dark areas as is the gray matter, or where the neurons are, and the lighter area is the white matter, where the tracks, and white matter tracks are important ingredients for communication. And you can say diseases of white matter tracks are typical, like MS and other demyelinating diseases. So what they found using this approach, again, and this is an independent study, okay? So this is, the first study was looking at genes, this study is looking at the brains. And not surprisingly, I copied the same diagram because the genes that were identified as being different between controls and patients were, again, genes expressed in neurons. And there are several, neurexin is NR XN1, and neurexin is important to communicate. So from the presynaptic side to the postsynaptic sides in the inset here. So, and that is also seen here in terms of layers. I told you about layers, and the neurons affected the most were in layers 2 and 3. There are 6 layers in the cortex where neurons are in different layers, and these layers in the 2, 3, which is where communication, intracortical communication from cortex, from one neuron to the side of the same neuron in the same side of the cortex. And then those are glutamatergic neurons, which are excitatory. And then we have inhibitory neurons with vasoactive intestinal peptide, VIP, are also involved. So those are the main neurons that were involved. So again, autism is a disease of neurons, particularly in the cortex, affecting brain development. So what else we know about autism? And we know the brains, the overall structure of the brain, and the function is different. So the next one, we are going to see that interesting, the children seem to have what we call accelerated brain growth. And again, if you can see in the first A, looks like the controls, the autistic patients in these red dots are above the normal curve okay, let me start all over. We are born with a brain size, and through we need to reduce the brain size to focus on the most important connections and disregards the noise. And this is something important in development. So what we look at here is, if you look at the B panel and look at the brain weight curve in the patients with autism versus control, you can see first that the autistic patients have higher brain volumes when they start with a younger age. Okay, so this is accelerated growth of overgrowth is at the beginning, but then the controls catch up, and then they are similar. But the problem of the overgrowth is when the brain is developing, so they are making excessive or inappropriate connections, and so too much connectivity could be good, too little connectivity is bad. So this is something that is critical, is to have the right number of neurons and the right number of connections and the communications between them. So in this, to talk about neurons, so not only the weights of the brain is different, but also the neuron numbers are different. And again, here you can see in different brain regions, in green is the prefrontal cortex, and then in the inside, this section inside shows you inside the brain where you will look at it. But in all of these brain regions, you're going to see that the patients with schizophrenia will have higher number of neurons. Oh, sorry, schizophrenia. I'm sorry, I apologize. That's what I studied most. So my expertise is primarily schizophrenia, secondary autism. So you can see that the number of neurons in different brain regions of the patients with autism are increased. They are always higher than the controls. And that's important to understand that. You would say more is better, but the problem with brain and with many things, more when it's not the right size and not the right connectivity, it's not good. So, and that's something that we'll talk more about connectivity in a minute. But so if you look at the cortex again, and this is, again, I want to emphasize in the cortex, another study show patches of disorganized cell layers in the neocortex. The neocortex is the cortex in the brain. And you can see here, what they do is they section the brain and look at the cortex using different markers of different type of cell types. And in adult, in child, what I want you is not to remember the names of the genes or the staining. Sneasel staining is for staining all neurons, but and the layers. But you can see that the surface is smooth within this range. You don't see any disorganized. So this is the control subjects. Now you go to the patients. Now you can see the Nissl staining just shows these breaks in the smooth, and then you have areas when the layers go up and down, and you can see it with all these markers, and also looking more in the false color, so you can see it in more detail, each layer, you can see each marker, and you can see there are areas where the layering is totally disrupted. So those neurons are not organized. In all these layers are not organized properly. And this is also important to understand. It's not just the frontal cortex that we were talking about. It's the frontal cortex, the temporal cortex to your side, the parietal cortex top to side, and then the occipital lobe to the back. So all of these are areas that have this disorganized orientation or localization of neurons. So if we were going to dig in more closely and look at each neuron individually, okay, in each layer, and sorry I didn't tell you, but I have copies if anyone wants copies of these papers or more explanation on any of these papers. I'll be happy to send them to you. So these are available through PubMed, and I assist, neuroscientists and scientists that get access to those. So I'll give you the references as well, so you can check them. So basically, what you are seeing here in A is neurons in two colors. In here, showing in the control and the patients, and you can see that in the patients, they have more of these spines. So this is a dendrite. Each neuron has several dendrites, and each dendrite, the sites of communication between one neuron and another is those spines, these little protrusions from the dendrite, is called spines, and then it's where synapses occur. So if you have more spines, so you have more synapses. And you can tell that in quantitation is number of spines per 10 microns, and you can see that the patients at all these different ages, they have increased number of spines. Okay, so that's the important thing, is that the patients have more spines, and that And indeed, you can see that while in normal subjects, the spine number decreases with age, if you compare the groups in the groups children 2 to 8 versus 13 to 18, you can see a dramatic drop in the number of or the spine density, number of spines to 10 microns. And in the children with autism, you see very small declines. So it's not helpful with age. Okay, moving on. What are the consequences of this increased synapses, increased neurons? And what is in decrease is abnormal developmental connectivity. So instead of we focus in specific areas, and the type of graph can be difficult to interpret, but each region is identified as a circle, and each hemisphere is so we have right and left, and you have you can see in the control you have several connections, but in the autism patients, they have much more connections in both sides, and they don't have much connection between one hemisphere to another. So it's a very disorganized. More connections, again, doesn't mean that the brain functions in a better way. And this was also evident. So in studies of fMRI, functional brain imaging, and basically fMRI is like an MRI machine, but it looks at the blood flow as it goes, and the more active an area is, the more blood flow, so we can look at which areas, it's not only the structure, but which areas are more active. And as I mentioned before, patients have problems with language, and there are two areas in language. We have the Wernicke area here in the superior temporal gyrus, and you can see it in one circle, and then the other, so the superior temporal gyrus communicates with the Broca area for language organization and articulation and speech, and then those areas need to be connected properly to be able to interpret and to articulate words or meaningful language. And the most important part in this is that if you see the synchrony, those areas need to be overlapped. The activity needs to overlap, goes up and down together, like you see in the bottom panel in the control. But as you can see in the group with autism, some area will go up and the other will go down at different times. So this is kind of lack of synchrony, and this is under connectivity or dysregulated connectivity. And this is in even in the high functioning autistic patients. And the same thing happens in the low function. They have the same very pronounced, but in this case they have increased connectivity, like they talk too much and they don't stop. It's like it's a constant flow, and the rhythm of this is also affected. And not surprisingly, if you have lot of connectivity between how one brain region connects to another brain region, you have also problems in the tracks, the white matter tracks that are associated with that communication. And again, it says again that they have more abnormal and increased volumes on those white matter tracks. And if you see the arcuate fasciculum, the uncinate fasciculum between, again, the areas that we mentioned before. So there are a lot of different areas of white matter tracks that are increasing volume because there's constant communication. The more communication, the more you have. And the problem is that because of this constant communication, there may be confusion because we have to have appropriate communication between those two regions. Okay, so far questions here? No. Okay. So we'll go Sorry, I can slow down if you need. I just realized that I wanted to explain things in details because it's important to know that it's nobody's fault, and it's important to know also that it's a disease of the brain. It's not the child's fault, it's not the mother's fault. So we'll talk more about that. But it's a problem of a disease affecting development, affecting brain development.
Dr. Bizzozero,
Yes.
it almost seems like the overconnection between different regions of the brain prevent those regions from being able to specialize in their functions. And so you may get a stimulation in one region that in a normal person wouldn't connect to a whole bunch of other regions. But with autism, you get one area activated and it immediately connects to a whole bunch of others because of these additional connections, and that then prevents each one of these specialized locations from being able to really do the function that it's supposed to do effectively.
Great question. Should I repeat to the audience?
No, I think they heard it.
They
Yeah.
heard it? They heard it?
Thank you.
Okay. So yeah, so let me see if I can go back. Sorry, I'll have to back. This is rats. There are connectivities. Remember when I said all the brain regions that have abnormal neuronal numbers?
Yes.
We are here. So it connects, it's frontal, temporal, parietal, occipital. So this is where we have this regulation. And this is also reflected in this,
Yes.
in a normal connectivity. And that's exactly what happens. If you have a normal connectivity, or you have more connectivity because more neurons and more synapses, then what you have is abnormal brain activation patterns, and then abnormal white matter tracks connecting this. And you can see all of the tracks, there are several tracks. All of the tracks that are in color are abnormal.
Mm-hmm.
Only the white tracks, the few white tracks that is hard to see, are not affected. So it's affecting the whole brain. That's a really good, great question. All right, besides that, what are the things we notice in the brain of patients? And I don't know if I've talked about schizophrenia and neuroinflammation and neuroimmune. I don't remember right now. But there are nowadays where we know that many of these disorders have also neuroimmune activation. So the immune cells that are resident in the brain called microglia, and those are like immune cells. They go there, and if they are cell debris, they will phagocytize. And this is important, not only the function, we need microglia, and we need those cells to what we call synaptic pruning. And that's what happens in here. The synapses don't prune. So if you have a tree with too many branches and so then if you don't prune, then things don't, in this case, will not be as effective. Mm-hmm. Will be a lot of noise, a lot of traffic. And so for a child, that noise and traffic will be difficult. For a patient with schizophrenia too, if abnormal, there's a lot of noise and not enough signal to noise, will be also difficult. And there are other disorders like that. Obviously, schizophrenia has, on top of that, the hallucination and delusion. So it's also another set of activation that is not proper, because they have misperceptions where they think and hear these voices. And anyway, in schizophrenia, we know there are neuroimmune activation, and in autism too. And this is two recent studies showing a normal, so what is a normal, the control brain and on top, and the brain from an autistic subject at the bottom. And you can see all these lights all these lighting areas means that there's a lot of inflammation. All of these areas that are light up. And if you were to look, so this is PET scanning, you can look at microglia activation with PET, positron emission tomography, and you can look at this. But if you have a brain from a subject, you can also look microscopically, do a staining for microglia, and you can see a very angry microglia in brown, and you can see, all of these microglial cells and astrocytes in green. So all these cells means in the panels to the right means that there are much more microglial cells activated. So it's an inflammatory disorder that is not normal. So again, and what we know, and this is also in common with schizophrenia, that maternal immune activation via virus, remember I said the winter birth, the flu epidemics, and some risk factors. They don't cause this, but are increased risk factors. So any infection in the placental infections, all these are known to disrupt gene expression and cause, in animal models, autistic disorders. And these are, so this is kind of the genes that are dysregulated matched with inflammatory processes. So we know there's neuroinflammation is a contribution, okay? So far we have some of the brain characteristics. And now I wanted to switch gears in terms of inflammation, and we are going to talk about a topic I haven't introduced last time, that is the microbiome. I don't know if you heard, the microbiome is, there are millions and millions of bacteria in our gut and throat, all over, inside and outside and skin. But what we know, the gut-brain axis is very important for brain and for the function of the gut. And I didn't tell you until now, but these autistic children mainly has high incidence of GI problems, so which I'm gonna go more about. So that's why this international meeting, what they were trying to look as and these are findings supported, related to Autism Speaks. They said the microbiome in autism-related GI symptoms and behavior, because the gut talks to the brain and alters behavior, the brain talks to the gut and alters the microbiome. So the first study, I think it was published 5 years ago, and here says distinct microbiome neuroimmune signatures correlates with abdominal pain. And again, this is something that we are going to talk more. Sorry, I have to 7 or 7:30?
Probably about 7:10.
Okay. Oh, I have to go very fast. Okay, sorry. Okay, so
7:15.
I'll do 7:15. Thank you. So basically, is children, if they analyze the gut from the children, they find that they have different bacteria, and that bacteria are releasing inflammatory cytokines like interleukin-6, and they contribute also to increase tryptophan, and tryptophan is the precursor for serotonin, and so that's kind of, so we have two hits, neuroinflammation and neurotransmission. So those two hits are important. So those one study. The next study tried to validate this in a different way for fecal transplants. You have heard with fecal transplants, they can do transplant. So in this case, they transplanted the fecal material from the patients to mice. I know it sounds horrible, but the mice But the important part is now we have a system in which we know we only vary one thing. Okay, the mouse are identical in any shape and form except this. So you can isolate what is the role of the microbiome. And so in this study, they show that the mice show this different autistic-like behavior, but only if they have the microbiome from the children that had autism. The others did not. So, and why is that? Well, the way they found is the cells in the gut, they have these different metabolites. One is called taurine, the other is 5-amino-valeric acid. And they said that there were decrease in the autistic samples, and then if they supplemented them, they corrected this. So it's not a treatment, but it points out to the importance of autism of diet. And I just want to briefly touch to this. Many of the children that have autism have food allergies and food intolerance. And for those cases, only those cases, they have this casein-free, gluten-free diet. So if a child has autism plus all these food allergies, then they said that diet, it helps social skills, okay? But it's only for those that have the food intolerance. So what else we know about autism? Well, we know environmental factors. There are a lot of different exposures, prenatal exposures to pesticide, air pollution was, and this is from the National Institute of Environmental Health Sciences. Metals, pesticide, other mercury, lead, so we know contaminants in the water as the mom is pregnant increases risk, doesn't cause, but adds to risk. So like genetics, this increase the risk, and they can increase the risk for twofold. Instead of 1%, will be 2% of the population. But also Tylenol. This is a very, very recent study that I gave you the link in there, that women, we also saw Tylenol safe, but now they said Tylenol doses is not as safe, and when they find it in the blood in the baby, they said it increases the risk for autism. Okay. Yeah, so I would say if someone is a mom, it has to be, nothing, all pure, really, no pesticides, no organic and pure air and clean air, clean water, act and organic. Okay, things that you can do to lower the risk, great nutrition, and the other thing is folic acid. Folic acid is a supplement that all moms get, but here it says the important thing, the sooner they start is the better, and if someone is wanting to have children, they start to have folic acid first, because it's only within the first months of pregnancy. Many times women, they don't know when they become pregnant. Well, today is 2 weeks, so it's better to prevent with folic acid supplementation and vitamins. What does not cause autism, and I want to be really clear with you, is not vaccines. So vaccination is very important because measles epidemics, the anti-vax movement. And so my question to you is, this study is meta-analysis 1.2 million children, and they said MMR, mumps, measles, rubella, does not cause autism. In 1 point, this is the largest study today. How many was patients? They have this faulty, I think, fabricated study by Andrew Wakefield in England. His medical license was revoked. His study was also withdrawn from the journal JAMA. Everything has to be, how many he has? He has 12, 8 of which were autism, 3 with measles, and 1 I don't remember. So it was not something that you could put a study based on 12 children and a study based of 1.2 million. You can tell the difference. And so I would say having a child with measles, it can be deadly. So, no to the anti-vax, vaccinate, and it's more, it's the CDC, I give you more references that says the same thing. These are one of the meta-studies, and said doesn't, I'll give you reference. Thimerosal that was as in a preservative in the vaccine does not cause autism either, so vaccinate, tell everybody to please vaccinate the children with MMR and other vaccines. Okay, what we know about the current treatments, I gave you a list of the current treatments that include antipsychotics, SSRIs for depression, so the first two are antipsychotics, the second two are SSRI. Methylphenidate is the drug used for ADHD. Then there are a couple other drugs, and they are different doses, different side effects, but they are not totally effective. So they wanted to get a better drug. And the problem, the challenges of clinical trials is it 10,000 compounds, and it takes so many years, like 15, 20 years to get something in the market, from which the start is 10,000 to 1. But lucky this year, we found the one. And the one, some of the keys, we know one of the social hormones, and there are the two oxytocin, which probably know as the empathic hormone. Vasopressin is also good for social skills, and they are, so this study was 20 years old, and that said maybe there's something in there, and the vasopressin. This year, 2 studies, one in children for a group in Stanford and one in Roche. The first author of the paper was a former postdoc in my lab for 7 years, Federico Bolognani, and just with Roche. And then good news, in both seem to be working well. So if you're going to check clinicaltrials.org, the study in children is nasal infusions of vasopressin peptide, and the study in adults with autism is this vasopressin 1 receptor antagonist, Balobatam, And this is a breakthrough therapy designation, is something that is available right away because so promising results. All right, so I have 3 more slides for you. Okay, so one, at the beginning I said autism, thousands of genes, small effects, but there are other conditions that are single gene. And I gave you a very long list. I'm gonna talk about the first 2. And it's Rett syndrome and Fragile X syndrome. And all of these, if you read here, all of them have autistic characteristics, all of them. I have, they have you don't have to read here, it's not going to be easy to read. I see you squinting, I know how hard it is to read. Sorry, I cannot read them either, so, but you have copies, they have copies on my PowerPoint. So Rett syndrome is a devastating developmental disorder. The girls start well, and they have developmental delays. The single gene is called MECP2. It's in the X chromosome. And because women had 2 chromosomes X, the boys are lethal because since it's in chromosome X, and if this gene is mutated in chromosome X, and because the boys have a Y, then the boys will not survive. So this is lethal in boys. But in girls survive with severe developmental problems of autism. And this, I'll show you next. What happens is normal on top is the normal development and maturation in which neurons connect to each other. This is in green is in excitatory neuron. We have modulatory neurons, VIP, the same one I told you before. And then these 2 are inhibitors as well, parvalbumin and somatostatin inhibitory neurons. And what you see without MECP2, or when mutations in that doesn't happen, and therefore the children, they don't develop, and they have very severe form of autism. The second one is Fragile X syndrome. Is again this is in chromosome X, and at the end has in site of instability where they have a lot of mutations. In one of these mutations is the Fragile X. Fmri is a fragile mental retardation 1 protein, and the symptoms are also changes in the phenotype, physical phenotype, severe in ID, in intellectual disability, and they have all these terrible syndromes, symptoms, and that's something that is very rare. So it's 1 in 4,000 males, 1 in 8,000 females. So fortunate is a rare disorder. All right, so I'm done for this talk, and if you want to contact me, Betty or Tom have my email, and I will leave the presentation with them. And from those that are in the other side of the camera, if you have questions, please contact me through them.
Can they ask questions?
Yeah, they can.
Oh, they can ask questions now?
One person says thank you so much, and thank you, Nami Santa Fe. So,
Well, that's good.
yeah.
Thank you.
Any other questions?
You have.
I have a question.
Yes, Betty.
So,
Another thing.
so it's a developmental issue.
It's
brain
total.
developing. So the baby's born, brain starts developing.
The brain starts developing before the.
Okay.
That's the thing. Yeah,
Okay.
before the baby is born.
So is there at some point that the brain is not developing and then it starts developing, or vice versa? So somebody could like start off with autism and then not have it, or vice versa?
Well, unfortunately, once the brain develops, and there are children that have later on, the adult onset autism is different from the autism.
That's what I was gonna ask too, about adult onset.
Yeah, the adults is usually they have, and it's adult onset, and it's totally different. It's not as severe. Obviously, the signs, and you can tell a baby when the baby doesn't. They have tests to look at the baby faces the mom, and if they don't have good eye contact with the mom when they are, you can tell there's something not functioning well. But the development, I mean, the neurons start developing. Sorry, I need to go back to tell you exactly if they have. I'm more familiar with animal models than human development. Sorry, I studied that a long time ago. So, but neurons don't develop from day one of conception. It takes, okay, so here we have the developmental pattern. So first neurons develop, and they don't have, this is about 8 or 10 weeks of gestation, okay? And then they migrate, so the first are born, then they migrate to the cortex, so you have migration, then the outgrowth of processes. So you can see all the time, and the critical time mostly for autism is the migration and the connectivity and synaptic function. So that's the most critical time. So that's the time critical period. And as you can see, this occurs before birth and then continues after birth. Yes, question?
I'm not sure if it's autism or something, some other condition, but there are some individuals, and I'm wondering if it's this, that they are, I guess, hindered in their behavior, their thoughts and everything, but are also really strong in some other area.
Yes, savant. Yes, the high functional autism, they have, and savants. okay, they have functional symptoms, they have better verbal communication skills. They are autistic, but they are not as severe with this repetitive rocking, and so the verbal skills are preserved. So when I started going back at the beginning, we were talking about the core symptoms of autism. Sorry, before we went to, so verbal skills are important, but delayed language communication. So those individuals that have, they still have impaired social interactions, not the language and communication skills is better, and they are maybe genius in math and other, so it can be, so there are certain functions of the brain that are preserved. It's very rare. I mean, most patients with autism have all of these three. But in very rare high function autism like Asperger, as the physician, the name of the physician, that characterize the symptoms of autism. In those cases, they have almost normal language and communication skills, but the other two, all of the others will be abnormal. So those two.
So the ones that are not really, really high functioning, but they're like creative in a special way, is there a high incidence of that or not?
No, those are the rare, very rare that some child we have all these characteristics, and then the majority, I don't know the incidence, I can check for you about how common, but
that's okay. I just wondered if it was rare or not that rare, you know?
Asperger disorder is 1/3. The incidence is 0.3 of the incidence. About 1/3. But then those that have high functioning, very high functioning in one area and not functioning at all in the other two, then it's very rare. So it's not the typical. It's not this typical. The typical classical autism. Yes.
When you talked about the hyperactivity of the inflammation system in the brain, the microglial coming through and being much more active, the Fmri or PET scans that showed the autism brain more inflamed. It seems that having all of those inflammation cells would actually go through and reduce the number of synapses because they'd be attacking things in the brain. But yet what you also said is that the synaptic pruning is less in the autism brain. And of course the synaptic pruning is important because
for normal
you
development.
want to get rid of all the synapses that aren't used very much so that the ones that are used can really develop to be very, very good.
Excellent question. And I thought about this. So let me tell you, I'll answer in two ways. The first way is that we know in schizophrenia is excessive pruning. Okay, so this is excessive pruning. And that we know some of the genes are associated with neuroimmune aspects, and also some of the genes, the complement, and I think I'll talk about that, but if not, I'll tell you there's a complement. The complement is, for coagulation, one of the factors. So the complement and for immune responses is the complements, not the coagulation factor. I misspoke in that. But anyway, those factors are important for taking care of the synapses during pruning. In the macroglia, what happens, I don't know if the macroglia really is the same type of microglia. It's not a normal function microglia. Maybe it's releasing, it's not pruning, it's more releasing cytokines and inflamed.
Okay.
Yeah, but I'm
It's
not sure.
pruning.
Well, if they are pruning, it has to be the microglia that are normal. So I don't want to spend too long. There are two types of microglia: the good microglia and the bad microglia. The activated microglia is kind of it's gone over all over the place, releasing factors, and the other is mostly for the basic aspects of the function that needs to be pruned at the level synapse. The other microglia is a different process.
Okay.
So there are two different types of microglia, different types of astrocytes. So, but it's true. I don't know the levels of inflammation in all the patients. I have to say maybe some patients have more inflammation than others as well.
Thank you.
Okay. Good questions. All right. I think it's
That's a lot.
a lot to digest, but I know that, I'll be happy to answer questions, so please let me know and please email me. And
Well, let us thank our
Thank you
speakers.
very much.
Thank you.